The part 4 of somaclonal variation. In this video, we are going to study the causes of somaclonal variation. Now, if we look at the PPT, okay, then we'll come to know the various causes. Now, physiological causes. So, what is a physiological causes? Okay, so exposure of culture to plant growth regulators and culture conditions. Now, please remember that we perform the all activity of tissue culture under very controlled condition that we feel are suitable for plants. So what we do, we expose this culture, we expose this plant tissue to various growth regulators, auxin, cytokinin, gibberellin acids, and these all growth regulators directly affect the DNA, because the role of growth regulator is to increase the rate of cell division, and hence these growth regulators generally act on a DNA, and while acting on DNA, these growth regulators brings about a mutation in its genetic makeup. So in natural environment, there are no such a growth regulators we are externally applied. They are present inside a plant, but while performing tissue culture, these growth regulators are there in the media itself, and hence they are applied externally to plants. Hence they bring about a changes. Then there are cultural conditions. What we do, we provide a solidified medium instead of soil. That condition is also going to affect the development of root. Moreover, instead of sunlight, we provide artificial light. We provide perfect temperature, that is 25 degrees Celsius. Okay, but in nature the temperature kept fluctuating. Also, these all the cultural conditions that we provide may bring for the physiological causes of somaclonal variation. Then there is a genetic cause. Genetic cause involves changes in chromosome number, euploidy, that is changes in number of chromosome sets, aneuploidy, that is changes in a part of chromosome sets, which further includes polyploidy and monoploidy. So genetic causes are due to what happens when plant cells start dividing. So they divide under the influence of growth hormone. So this division may lead to the abnormal cell division, and due to abnormal cell division, there is a generation of changes in the chromosomal variation, and it includes euploidy, aneuploidy, polyploidy and monoploidy. Moreover, due to this abnormal cell division, there is a change in the chromosome structure, that is deletion, inversion, duplication and translocation. So these all factors leads to the changes to the generation of somaclonal variation. Then there is a further genetic causes, that is gene mutation, that is transition, transversion, inversion, deletion. Then there is a plasma gene mutation, and then there are transposable elements. So genetic causes further includes the changes in DNA. Okay, so now the what we say, there is a detection. So we have to detect this what we say, the somaclonal variation. So deletion of altered fragment by using the restriction enzyme, changes in protein, loss or gain of protein band, alteration in level of specific protein, methylation of DNA. Okay, so basically what is happening now? So moreover, due to the cultural condition or various physiological factors, the whole genetics or of the cell is getting affected. Somehow the DNA is getting affected, DNA methylation pattern is affecting. So DNA methylation is responsible for the compact binding of DNA inside a nucleus. So this pattern is getting affected. Okay, then there is a changes in a what we said, sequence of DNA due to mutation. Then so that will obviously lead to the changes in the restriction fragments. Okay, so overall these causes are leading to the variation in the genetic makeup of plant. So these variation in genetic makeup of plant will lead to the production of somaclonal variation. Okay, now if we look at the biochemical causes, so what is the biochemical causes? There is a lack of photosynthetic activity due to alteration in carbon metabolism. What happens in nature? Plants are autotrophic. Please remember that plants are autotrophic in nature, but when we subject these plants to in vitro condition, then these plants become heterotrophic. So heterotrophic mode of what we say mode leads to the alteration in photosynthetic activity. Then there is a biosynthesis of start via carotenoid pathway, nitrogen metabolite, and antibiotic resistance, and antibiotics which are using for the tissue culture. Okay, remaining causes will be converted will be discussed in